explicaré serà una miqueta el que ha dit el Joan i també uns projectes nous que tenim en l'àmbit de la fenòmica. So he told me to make the talk in English, so I will do so. Basically, in my program, we are interested in the relationship between genotype and phenotype, and mainly in human complex disorders. And in my lab, we are specifically working in trying to determine in animal models what is the impact on higher cognitive and behavioral aspects of different alterations at the gene regulation and interaction level. So the idea is that we identify, the program identifies some possible candidate genes for different disorders, and then we go into the animal modeling and into the different levels. In my lab, we also go back there, so we are also interested in how this alteration at the higher function level is correlated with structural or functional alteration at the system levels and also at the cellular level. So I have structured the talk in a couple of different topics. In the first one, I will try to propose you that we have to change a little bit our concepts in how we do this functional annotation in mouse models. So that what I'm proposing is that, in fact, a different concept in mouse phenomics will be very useful to identify biosignatures for predisposition, prodromal period, and also for expressed disease. So the advantage of this is that we will be also able to determine some aspects that are necessary also in human disorders, like the natural history of the disease, which are the biomarkers that help us to predict severity or response to treatment, etc. So this is basically what we will talk about. And just to convince you that mice are good models for central nervous system disease, in fact, as you know, brain structure is not as different as you might think, but also the phenotypic outcomes are similar. The phenotypes that we can measure in mice are very similar to those that we can measure in humans. Of course, we cannot study some things like language. We have to cope with the differences in the ethogram. But even so, the phenotypic screens have been able to identify a role for some very important candidate genes in some neurological and neuropsychiatric disorders. So, because I didn't know how aware you are about how we do these kind of experiments, I will make a little tour about around some of those. Okay, so first thing that we have to do, of course, is diagnose our mice. And this is very important because, in fact, mice in this context are in vivo models for function annotation. And this will determine how we understand the function of a particular gene or of a particular alteration at the genetic level. So this is very important, how we annotate these phenotypes. Kind of apparatus we use are very variable, very different, and in some cases we have to use our imagination to create them. But those things allow us to make studies in different functional domains: emotionality, motor activity, learning, sensory systems, etc. But also help us to introduce some environmental modifiers into our models. So we can analyze the effects of environment, the effects of stress, or the effects of enrichment, environmental enrichment, and I will propose you some examples of that also. Okay? Apart from having different, like, general functional domains in our analysis, we can also have disorder-specific tests that help us to study specific phenotypes. Okay? So these are just examples. These are very simple tests, but those help us to make a general neurological diagnose. And we can also use, for example, disorder-specific tests. This is called the mouse defense test battery, and is helpful to understand to what extent our mouse model is expressing some of the components of the panic response. So basically, well, this is me after the mouse with a rat. This is a natural predator of the mouse. So this test is sensitive to, all of those are pharmacologically validated, but this is sensitive to panicogenic and panicolytic agents. We can also study other things like learning and memory. And for example, here I propose you one of the tests we use for studying recent memory, which is important, for example, in models for Alzheimer's disease, as you know. So here we put this mouse on the platform, and when he steps down, he receives a little shock. You may think it's not so little because he will jump, but it's not very strong, I tell you. Believe me. And so if the mouse learns that he will receive a shock and he's able to retain this information, then 24 hours later, what we will see is that he will not step down, which is a natural tendency for the mouse. So he will have to inhibit a natural tendency to step down. Okay, so just pay attention. This is what normally a mouse does. When you put him in a platform, he receives here the shock. Okay, and then 24 hours later, you see that the latency to step down is much longer. He remembers, even though, of course, he will, at the very end, step down. Okay? So this is a nice proxy, behavioral proxy, to study this recent memory. Another very well-known test has to do with spatial memory. Why spatial memory? Because this model is considered as a model of, let's call it, higher cognitive performance in human. Why? Because it requires what has been called the generation of internal representation. You need to create a map to locate something in the space, okay? And this has been correlated with mainly the hippocampal function. So how do we do this test? What we do is we use this pool, because mice do not like water as kids. I don't know if you have children, but it's exactly the same thing. So you put an escape platform there, but it's not visible. It's under the water level, and the water is stained in white, so you cannot see it. So basically, what the mouse has to do is to create this kind of map, because you will introduce it into the pool through different entrance positions. So it's like if you go to the Sagrada Familia and they put you first in Diagonal and then in Meridiana and then in Horta and in the port. So you have to create a map and search for visuospatial references that help you to locate the platform. This generates a learning curve. So this is the distance to reach the platform, and it's being reduced along sessions. And here, this is a wild type mouse. So at the first session, he acquires the task, and he reaches what we call the asymptotic level of execution. Here is a model of Down syndrome, which is one of the disorders that we are studying. And what you see is that these guys do not learn in the first sessions. Then suddenly they get inspired, and they need double number of sessions to reach the same execution levels as the wild types. So here we have the example of a wild type mouse that is in the first session when he has already learned. You see, he reaches the platform and is able to escape. And this is as a comparison. Here we have this trisomic model in which you see that it's not only that it takes much more time, but it's also that he's using strategies that are non-spatial strategies. He's not efficient in searching for the platform in a convenient way. So, in fact, we can analyze these by looking at how the stage in the different areas of the pool, and the platform should be here, is evolving along time. And we have developed a proxy for that, a plugin for that, so we can see that in this case, the wild types are searching for the platform very efficiently. Okay. In general, what we need when we have a mouse model of a disease, we need some validation criteria. What does this mean? It means that the mouse model that we are pretending to be a model of a disease has to have some validity criterion to be covered. So basically, what we call face validity, this means that the behavioral response that we observe in the mouse model should be similar or identical to that observed in human patients. For example, if you are creating a model of anxiety, it has to be anxious. Then the predictive validity, if this anxiety has the same grounds as in humans, then these mouse models should be sensitive to clinically effective pharmacological agents. And finally, this is more difficult, the construct validity. So this relates to the similarity between the theoretical rationale that underlies the mouse model and the human behavior. For example, if you are creating a model of Parkinson's disease, you should see a degeneration of the dopaminergic system. Okay? Okay, so then when you do all these tests, all this test battery, then you will see that some tests are done better or worse or equal or different from your wild type, reference wild type mouse, and you will diagnose your mouse, okay? You will see that finally you have whatever phenotype. The problem is that sometimes this is not perfect. What do I mean? This is what we have been working during many, many years, what is called phenotype ontology. So to give a real, let's say, concept behind the phenotypes, to interpret the phenotypes. For example, I put you the example of schizophrenia because it's a very difficult example, okay? And it's not very well done. So, for example, for schizophrenia, as marker of the positive symptoms like psychomotor agitation and hyperresponsivity to psychomimetic drugs, but this is okay, but they use as an indicator of psychomotor agitation the locomotor response in a novel environment. And this can be very unspecific. You can find hyperactivity in many, many models. So this is not specific. As for the negative symptoms that you have here, like, I don't know, blunted affect, social withdrawal, and loss of pleasure, we use things like the sucrose preference test, memory in the Morris-Water maze, or the purpose inhibition that can be affected also in many other things, okay? So basically, the mouse orthologies for the major symptom categories are not really the best, let's say so. Okay, so then this comes in part because we are trying to express behavioral domains in the mouse that are not always easy to see. And in fact, what we know is that the behavioral profile in mice and in humans is a mixture, is a variety of interacting motivational systems. And what we try in most tests is to focus on one motivational system. And this is one of the reasons that limit our ability to really reproduce things. On the other hand, when we use this standalone battery of tests, in fact, we are not putting the animals in the best environment to express the behavior. So, in fact, things have to be done in a biologically relevant setting, okay? So, then, how we want to try to solve all these problems. Not meaning that these tests do not have their value. I mean, what we want to do is to mix both things. But basically, the final idea is to be able to translate this into clinics, of course. But what we want to do is to use non-invasive methods in order to be able to interface these basic and clinical findings. So basically, the idea is to be able to identify novel and multiply interconnected behavioral markers, to delineate the critical periods for disease initiation and progress. If you just take your mouse when you have the symptoms, as you do with the patient, you take your patient when he or she has the symptoms, and then you try to understand what is going on. But what is going on at this stage is probably not only the cause, but the consequence of what has happened across all the disease. So what is important is to try to see the whole picture from the beginning. So, of course, this means that you will have to analyze also what we can call the prodromal period. And also, this will help to, hopefully, to follow up the treatment efficacy. So from the mouse point of view, this requires a whole technological implementation, okay? This is not in place by now. So this means that we need to implement, and we are doing this by now, an interactive room that will be the home cage of the animal. They will be in social groups, so we will have transponders, identifiers for every mouse. And this will allow us to follow the evolution of the mouse behavior, but also it will allow us to modulate the environmental conditions, to manipulate different environmental aspects to see what happens, okay? So this is what we call the intelligent environment, smart. And the idea is to there use some tests that are disorder specific, but also then to do the other level measurements like brain molecular and cellular changes upon different changes during the normal disease, but also upon challenges. So the final aim is to try to find accessible biomarkers that are in mice expressing something that is comparable to brain, and then try to propose this as a proxy in humans for the CNS function or reactivity. Okay? Among others, we are already measuring some things like the real activity, and I will show you some examples of that, but also other things like, you know, you can do telemetry in these guys, in mice, but you can also measure biomarkers in the breath condensate like cortisol, monoaminotic products, etc. Of course, this needs also a technical implementation. So, you may think, okay, this is very nice, but do you think this can work? This is very complex. How would you do this? Well, we have started with some experiments using this model, kind of more easy model. But basically, I will explain you a history that we are now working on, and is related to mouse models of eating disorders. As you know, there are several genetic models. I will not go into details of all of the models that have been created that show obesity and eating alterations and so on. And there are other models that are behavioral models. So, for example, the exposure to high-fat or palatable diets like cafeteria diets, high sucrose diet, the Western diet, etc. Okay. The problem of all these studies is that very few of those have looked at the longitudinal level, have used this as a longitudinal analysis model, and very few have explored a choice behavior. So, what we decided, well, these are the shortcomings, sorry, I forgot that I had that. The problem of those tests, of those models, is that they are short-term, as we were saying before, no? Standalone short-term measurements that may not be functionally relevant to long-term changes, in this case in body weight, but we could speak about behavior also. The measurements of eating in weight-stable obese individuals are not informing us about the developmental phase when they get obese, okay? And finally, it's important to identify these eating patterns that are consistent with the obesity susceptibility before the obesity develops, okay? So, just to make a very short introduction on that, in fact, what we were interested in were in the compulsive elements that are accompanying the development of this overfeeding obesity. So obesity is a pandemic now, and this means that we are probably facing something that is more environmental, let's say so. So, in fact, what has been shown is that this compulsive food intake shares some common mechanisms with drug seeking, as you know, and there have been some publications in the last years about that. And this includes the endogenous reward and memory system, no? Which reactivation will promote these motivational states for compulsive seeking food-taking behavior. So what our approximation, our approach was to induce obesity by promoting, by using a diet in which they can choose, the animal can choose between a highly palatable food like the chocolate and standard chow. The control group is just standard chow, okay? And the idea was to characterize the short and long-term appetite behaviors in the control and the chocolate-exposed mice, and see the long-term body weight regulation and also the key components in eating behavior. Okay? And we use this initial model for our tests. So this is a high-resolution recording system in which what we basically assess is the fine structure of eating and drinking with a specific software, and also the activity. Okay. So basically, I will explain you a little bit the technical details. What we can measure is how the food intake takes place, and the drink intake also, and then we can correlate these second by second with the activity. So for example, what we saw is that for each bout of eating, we could observe a reading activity because the hoppers are a little bit high. So it was very well correlated. So basically, what we have, as I told you, is these two situations. Of course, sorry, the energy intake per 24 hours is increased in the obese mice. Okay, here you have the data. But what we see is that there is a clear preference for the chocolate versus the chow. So there is a rewarding effect. As you can imagine, this is not very surprising, no? They like chocolate. Everybody asked me, why don't we use cheese? They like more the chocolate than the cheese, I must say. Okay, so anyway, here we have the graphs for that. And of course, they develop obesity. Here we have, this is the developmental phase. So the idea was to look at the behavior, how the behavior changes during this developmental phase. Okay? And one thing that we do is what we call an advanced meal pattern analysis. This includes time courses of access to the meals, to the food, duration and recurrence of intervals of eating and drinking acts, circadian and ultradian periodicities, cross-correlative relationships, and also the event-related frequencies. Okay? So just by eye, you can see that here is our control, the standard chow mice, and here we have in gray, this is the night period. So the night period is the active period in rodents. These are nocturnal animals. And what you see is that they have these eating periods here, okay? And if you look at our chocolate mice, our bees mice, this is, I think this was in the second week after exposing them to choice. So you can see that the structure of the feeding behavior is completely changed. In fact, they are snacking. They have very little bouts, okay? And this is interesting because unlike feeding behavior, which is quite stable and stereotyped, feeding clusters, these little feeding bouts, reflects the animal's motivational status. So that the number and the duration show variance in accordance with factors such as palatability, for example, or such as satiety instead. I think satiety, I never know how to say this word. So basically we had to define what we consider about, and we did this looking at the temporal distribution and the pauses between feeding acts. And this helped us to look at the whole feeding behavior. So there are a lot of qualitative features of feeding that are characterized for a sequence or a structure of actions that are normally driven by the need to meet nutritional requirements. But this is not the case. in our mouse, because the feeding bouts are more driven by motivational stages. Okay? So what happens here is that we are facing a kind of non-homeostatic eating. Okay? Their meal size and frequency and satiety are not driven by meeting the nutritional requirements. And what happens? What happens is that then, as you saw before, the number of meals are increased. So this is the chocolate meals, and this is the standard chow meals. Okay? But look at the meal size. The meal size is not increased, it's decreased. Okay? So the number of feeding acts is increased, but the size is decreased. So they are, as we said, no snacking. Moreover, we wanted to measure also how the factors, the elements stopping or initiating feeding were affected. So to do this, we have to measure the interval between feeding acts, but also what we call the satiety ratio. Okay? So this is the amount of energy they need to get satiated. And what is interesting is that, in fact, the amount of normal chow that they have to eat to get satiated is increased. Whereas in the chocolate, it's decreased or normal, let's say. And this is something that has been shown in eating disorders in humans that exhibit disturbed satiety responses. Okay? So the idea was then to look at the eating rate, how fast they eat. And the normal chow, they eat it normal, but look at the chocolate. The chocolate, they eat it very, very fast, no? And this is very similar to what happens during binge eating episodes, no? In which patients often describe this rapid consumption of food. Okay? And also this has been described in bulimia nervosa patients. Okay, so then we decided to put them on diet. Okay? So we limited the access of these guys to the chocolate for 1 hour per day. Okay? And we counted the numbers of meals in 1 hour. So this is what happens during 1 hour in the controls. This is the number of meals, so less than 1. Here is the number of meals in the obese, in the baseline, with the chocolate available. They normally eat much more chocolate than normal chow. This is why you don't see the gray bar. But look what happens when we return the chocolate for 1 hour. In this hour, they eat the same amount of chocolate, the same number of meals of chocolate, than they do in 24 hours if they have the chocolate available. So they are showing this compulsive behavior. And in fact, this happens also when we starve the mice, when we don't put neither chocolate nor normal chow. And here, also something interesting is that when we only limit the access to chocolate, is that they don't compensate the energy intake by normal chow. So the salad bar doesn't work. Don't make salad diets because it doesn't work. Okay, no, it's just joking, but it's true. They are not able to maintain the energy intake in absence of chocolate. So this is indicating us some inflexibility in their behavioral attitude. So, what else? We can do something else. We can see to what extent they are able to avoid or to work to get the chocolate, okay? And to avoid, for example, dangerous situations. So, to this end, we have used an open field. And in this open field, the normal behavior of rodents is that they avoid open spaces. Okay, so if we have a rodent here, it will not be there in the center of the room. It will be here or hiding somewhere. Okay, because they need to get protected of the predators. Okay, so normally in an open field, the mice, a normal mouse, would avoid the center. So we put the chocolate just in the center, exposed. Okay? And what we see is that, in fact, here you see, yes. In fact, the time at the chocolate and the number of snacks was pretty much higher in our chocolate mice, in our obese mice. So they're able to go for the chocolate even though everything is dangerous, and they fight for it, avoiding this fear. So mainly, from this part, what we can see is that, in fact, this is they gain body weight, etc., and the meal pattern analysis revealed some circadian changes and feeding structure alterations. As you see, the reinforcing and rewarding effect of the chocolate is reflected by the preference, and the anticipatory place preference also that we did. And the compulsive elements are shown or reflected by the maintenance of chocolate eating despite, for example, adverse bitter taste caused by alteration, or in the open field, as I show you, seeking the behavior in this aversive situation. And the inflexibility is reflected by neglecting the chow when the access to chalk is seized or temporarily limited. But of course, we wanted to see what happened in the brain of these guys. So first we did a PET study. And just by using glucose consumption, we could see a very important change in their brains. Not only that, but we also saw differences in, I will not go into all the details because it's very long, but just to explain you the concept, differences at the dopaminergic level, basically. And the problem is that we have all this behavioral data. You can imagine strings of data because we record the activity and the feeding behavior for 7 months every day, and the sensitivity is around 1 second. So it's a lot of data. So we have a lot of information, but we need to understand what of these changes in behavior could be a good marker of the changes within the brain. So to try to understand this, what we do is a model. Not this one, but a model. And this is difficult because there are not too many developments in working with this very long-term longitudinal information. So the idea was to, in fact, we are doing this in collaboration with a group of Cedric Notre Dame in the CRG, in the bioinformatics program. So the idea was to try to identify changes in the patterns, in these patterns that we have, to try to understand when we go from state 1 to state 2, which will be predictive of the development of all the alterations in the behavior. Okay? So to this end, they proposed to use what is called the hidden Markov modeling that is able to identify subtle repetitive patterns. And this has been used, for example, to predict an unfair dice, no? You throw it, and this system can predict which is the unfair dice. So we had to decide which of all the parameters we were working with could be interesting. So basically, we had all these observations, the feeding parameters, like meal duration, intake amount, etc. And the hidden states will be this feeding pattern. But then we had also this activity recording. So what we decided to do is to measure the transitions. So we have the transition from normal chow to chocolate, to water, again to normal chow, and this can have all the combinations that you can imagine. And in fact, we were able to cluster this information into bins, and just by eye, you can see the difference between these cages of the lean mice and these cages of the obese mice. And what is in blue is the transitions between the standard chow and the standard chow. So going back and forth to the normal chow, okay? But what is even more interesting is that here we can see already some outliers. So there are some lean mice that show a more obese profile, and vice versa. We have some of the obese mice that show more this lean profile, okay? But even more interesting, when we analyze this data, what we could find was a correlation between these transitions from standard chow to standard chow, okay, when we had only the mice there for 11 days, so they had not even develop obesity. They were just on the beginning on the test. So at 11 days, these transitions are correlated and predict the relative weight increase 6 weeks later. Okay? But even more interestingly, there is a negative correlation between these transitions at 11 days and the changes in the dopamine receptors type 2 6 months later. So we are able, with this parameter, to predict which mice will undergo the modifications in the dopamine 2 receptor. Okay? So again, just to remind you, the first 11 days predict the weight intake after 6 weeks, but also the dopaminergic receptor changes after 6 months. So the idea is also to do this in parallel with humans. And this is even more difficult. But we have already several technological developments that are allowing us to do this, the measurements of activity, et cetera, et cetera, et cetera. And we can discuss a little bit more after, but just to show you some examples. For example, here we have the activity recordings in humans and in mice, okay, which look pretty similar, as you see. And here is a healthy control, healthy human volunteer compared with a bipolar disorder patient. And here is the wild-type mouse compared to that knockout, dopamine receptor, dopamine transporter knockout mice. Okay, so it's not too bad. Promising. This means that we will have also to look at the activity measurements by themselves and try to find out from all these different trajectories, different tracking files that we have, what is the typology, what are the behavioral strategies that the mouse or the person are using. And the problem is that we need to identify these. And in fact, we are proposing to use artificial neural networks to try to identify these patterns. And how do we do this? This is not simple. This is very complex, because first we have to smooth the data, because it's a lot of information. These data are taken 0.1 second by 0.1 second, so it's a lot of data, really. But we cannot smooth it too much, because this is not very similar to this, or this to this. If we smooth too much, if we use too many iterations, finally we have a nonsense behavior, not representative. So basically what we are doing now is to try to develop this system. So we are capturing the path orientation, the angle, also we are binning the data, and we are adding to that other elements like speed, direction, and this in a dimension, in a temporal dimension, to see how this evolves across time. Okay, so we can have as many layers as we know, and then it's just a matter that of computizing that. So we get all these strings of data that are revealing a trajectory. Okay, and that help us then to be able to modelize all this. Okay, I don't know how. Is it very late? It's okay? Okay. 10 minutes. Well. Okay, so you have seen more or less this general approach that we are trying to develop. And that we hope we will be able to translate into humans. But, of course, every time that you are working in a longitudinal analysis, you have to take into account the environment, because the environment is very important. And we are very much interested in the gene-environment interactions. And in fact, I am very much in favor to Restrepo that said last year that scientists often assume that there is little impact of differences, for example, in housing environment on the data that they acquire. But this is not true. And for example, there is some studies in which they see how differences in the environment result in different levels of aggressivity in males, no? So it is very important to take into account, for example, here we see the mean frequency for fighting in a mouse that has been acynated or not. So it's very important. So this is something that we want to explore, but we want to explore it not only to have a control on it, but also to use it as a way to increase the efficiency of drug therapy. So basically, we want to see the effects of nurture and epigenetic changes on drug therapy efficiency. And I will just put a couple of examples, one example, of one of the models that we are using, that is the environmental enrichment model, just as a proof of concept that this really makes a difference in some cases. And as you know, we are working mainly with mental retardation models, okay? So basically what we use as an environmental enrichment model is the classical one, in which you use different stimulus that are targeting different systems in the brain. So it's an overall stimulation. And it has been described for many years that this creates changes in learning and memory, so it increases learning and memory, and also it produces changes at the cellular level in the brain. So basically it increases the branching of the neurons and the number of spines, so the communication areas of the brain, so of the neuron, sorry. So what happens in mental retardation? In fact, as you know, we are working, I will just make you a little drive through the project. We work in Down syndrome, and we are interested in the therapy in Down syndrome. And what we know is that, in fact, these early intervention programs are able to really improve very much the social and cognitive capacities. But the problem is that these improvements are limited, as you know, and temporary. And this suggests that the activity-dependent plasticity is probably impaired. So we wanted to understand, what happened in a mouse model of Down syndrome, if this was really true, if it was the structural plasticity that was not taking place. And these were studies that we did several years ago. Here you have the same curve I showed you before in the Morris-Water maze, the learning curve in the control mice, you remember that? And here you see the alteration in the trisomic mice. This is a partial trisomy of the chromosome 16, that is the homologous to chromosome 21 in humans. And what we see is that when we enrich these groups, what we see is that both the controls and the trisomics get better, so the curves are displaced to the left. And in the case of the trisomics, already at the first session, they get the same asymptotic level as the wild type. So the phenotype is mainly rescued, okay? But the problem is to what extent this drives a structural alteration. And we know that the trisomic mice, as happens also in patients, they have alterations in the microarchitecture of the pyramidal neurons. And here you can see, these are horizontal slices in which you can see the whole basal dendritic tree. And what we see is that the whole area is reduced and the complexity is also reduced. This is quantified here. But also the number of spines is significantly reduced, of dendritic spines. So what happens upon enrichment? What we saw is that when we quantify the number of spines, here in the wild types you see a very nice increase, and this is 7 months after doing the treatment. But what happens in the trisomic is that, in fact, this structural plasticity is not taking place. So there is a lack of a stable structural trace of this experience-dependent neuroplasticity. And in fact, what happens is that the effects on behavior are not stable. They are fading away across time, okay? So, of course, the problem here in this trisomic mouse is that we have a partial trisomy. This means that we have a lot of genes there. So this is a problem if you want to get for therapeutic targets. So you need to have something more specific. You cannot normalize the expression of 200 genes. That's impossible, okay? So the strategy was to try to find candidate genes that were able to explain this lack of stabilization of the environmental enrichment in Down syndrome. And one of our best candidates is called Dierssen. This is a kinase that is involved in different functions, basically in proliferation and differentiation, synaptic plasticity, but also in neurodegeneration. So we have, in our program, Christina Fillat created a transgenic mouse models, and these guys were only overexpressing Dierssen. Okay? So this is just to show you that the levels of overexpression in our transgenic mice are very similar to those observed in Down syndrome brains and in the trisomic mouse. So we have similar levels of overexpression. And to make a very, very long story very short, I have to say that increases in Dierssen dosage create learning problems, but also affect the structure of the pyramidal neurons, as also happens in the trisomic mouse. And in fact, this is what we call the dosage-sensitive gene. So we need the correct amount of DIAC to make the job, because also if in the heterozygous mice, we see these alterations. Okay? So the first thing that we needed to know was if the environmental enrichment was also able to rescue the cognitive alterations in transgenic mice, as it happened in the trisomic. And here I present you a different task. This is an object recognition task in which the mice have to recognize a new object that is put in an open field. And what we see is that the discrimination index is significantly reduced in transgenic mice, and this is work of Maria Martinez de la Gran and Meritxell Pons in my lab. And this is completely rescued by environmental enrichment. As also happened, and I showed you before, in the trisomic, the environmental enrichment was able to rescue the phenotype in the Morris-Water maze, so in the spatial learning task. And here is the curve from the transgenic, non-enriched, the wild types in blue, non-enriched, and here you have the enriched groups in which you see a complete rescue of the phenotype. Okay? So environmental enrichment is also producing this rescue of the phenotype in transgenic mice. But even more importantly, this is an experiment that we did by crossing TIE-YFP mice, so mice that have fluorescent pyramidal neurons, with our transgenic. So this is a double transgenic, and what Meritxell was seeing is that there is a complete disruption. This is a striatum radiatum in CA1 region. And you can see the complete disruption not only in the pyramidal layer itself, but also in the branches of these neurons. And if you compare it to the wild types, you see the more organized layers of cells and then the projections. And here you can see how this structure is rescued by environmental enrichment. So environment has a real impact and can be used as a tool for rescuing the phenotype. Okay? So the idea would be, in our case, to tailor this environmental manipulation to be able to activate selective brain circuits and devise a new route of performance prevention and treatment. Even more interestingly, we thought, okay, let's normalize Dierssen expression levels or activity levels in these Down syndrome models. Okay? So to this end, we use some inhibitors, and it has been described that the epigallocatechingalate, that is a polyphenol of the green tea, is able to inhibit Dierssen activity. And this is very nice because it's something that you can use also in patients. So the idea was to treat the animals either with water or with the tea and see what happened. And here you see again the object recognition task, and you see that here you see the impairment in the transgenic, and you see that this is recovered by the green tea. Interestingly, if you reduce the activity in the wild types, you get an impairment, but you don't need to avoid drinking green tea because these doses are much higher than what you can drink in a day of tea, so don't worry. Okay? So, not only that, but this green tea extract was also able to, here we have again, wild transgenic and wild types here with water. And with EGCG, with this polyphenol of the green tea, you see the recovery, again, of the phenotype in the transgenic. And here, it was no effect in the wild. So, good. Something is going on then. If we normalize the expression or the activity, in this case, of TOC1A, we can rescue the phenotypes. But could we maybe have a synergic effect? I mean, if we normalize TOC1A activity, would we potentiate the effects of environmental enrichment? So we did the experiment also, and what we found was, this is a complex thing, but I will drive you through. So here we have again, transgenics non-enriched, wild types non-enriched. You have seen this before. Here we have the enriched groups in green and in this purple here. This is the transgenic enriched, the wild type enriched. And here we see the transgenic enriched plus treated with green tea. If this is the effect of enrichment, look at the effect of enrichment with green tea. So we have a potentiation of the effect. Okay? So, as this is so promising, we went into clinics, because the idea is to go back to clinics. And we did some assays in Down syndrome individuals. This is just a pilot, and we had many problems that I can discuss with you later on. Because we wanted to stratify the study, so the groups were not very homogeneous. Pero bueno. And we had a placebo group and a EGCG treated group. So we had some neurological evaluation, but also we used some biomarkers that we can also discuss. I don't have time now, but we can discuss it later on. So basically what we saw is that there was some subclinical effect. It was not a huge effect on the test that we did, which, by the way, were not the best, because the battery, the Cantab battery that we were using is not tailored for mental retardation patients. But anyway, they had an effect that was significant and that was reversed after ceasing the treatment. But even more interestingly, we made the parents guess which was the treatment that their kids were receiving. And what we saw is that more than 90% of the parents were able to correctly guess if their kids, if their sons or daughters were on EGCG or on placebo. And the EGCG groups reported changes, visible changes, in many domains that we even didn't dare to test, like language, because we thought, well, language is very much impaired. We don't expect to see a change there, but the parents saw. And even things like independence, increased independence in this patient. So it was really very, very successful. So, and in fact, we are receiving a lot of complaints because we don't have money to make the second pilot. So the parents are calling us to ask, when are you going to follow this? Okay, so as a general conclusion, because I think I have run out of time, I hope I have convinced you about the usefulness, not only in basic research, but also in the translation into clinics of this next generation mouse genomic approach that we want to establish. And of course, we have discussed the positive aspects. We can analyze and use gene-environment interaction. We can do this longitudinal analysis that will help us to understand the natural history of the disease and hopefully to identify some biomarkers that can be useful to predict the severity and also the treatment outcomes. Of course, we will be able to see the impact of environmental regulation on specific genetic risk factors also. And this will help us also to perform some hypothesis-based preclinical therapeutic trials, as we have done for the Down syndrome. So, of course, withdrawals may be the opportunities for new prevention, new interventions, but we need to establish informative parameters and real environmental therapy. So this is still not there. And just to thank the people, basically the people in my lab that have been collaborating in the studies are Ignacio Saun, that is here, and Jerome MacDonald, that is now back in Cambridge, but they did the experiments on the obsessive-compulsive model. And then Maria Martinez and Mary Chale, that is here, are the persons that have done the experiments on Down syndrome. And we are collaborating with a group of Rafa de la Torre, and Maggi Ferrer and Susana Sola are also very much involved in the study. And of course, the people from the Jérôme Lejeune Foundation, and basically the people of the Fundació Catalana Síndrome de Down, they have been very, very helpful in helping us to recruit the patients. And thank you very much.